In this tutorial, we'll view two video animations of ELISA techniques. The first video is titled Enzyme-Linked Immunosorbent Assay to Measure Specific Serum Antibodies. And this video demonstrates an indirect ELISA assay. The second video is titled Enzyme Immunoassay to Detect Antigens. And the second video demonstrates a direct ELISA. So know that sometimes you'll see slightly different language to describe the assays, but for the purposes of our lab, we'll refer to them as indirect ELISA and direct ELISA. So let's take a look at the videos.
The enzyme-linked immunosorbent assay, or ELISA, is a commonly used format for serologic testing. The purpose of this animation is to explain how this test works. ELISA serologies are usually done in multi-well microtiter plates so that dilutions of serum can be easily prepared and tested. To better understand how this assay is done, let's take a closer look at what happens in one of the wells of this assay plate. To perform the assay, the wells of the plate are coated with the antigen of interest. For commercial tests, this would be done by the manufacturer of the assay. To begin testing, the wells are filled with dilutions of the patient's serum. If antibodies against the antigen are present, they will bind to the antigen fixed to the bottom of the wells. But only antigen-specific antibodies will bind to the wells. The wells are then washed out to remove all unbound antibodies. Next, a solution of an animal antibody against human antibodies is added. This second antibody is covalently conjugated to an enzyme. The wells are washed again, this time to remove any unbound enzyme-conjugated antibody. Finally, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate with the enzyme on the second antibody generates visible color. The development of color in the wells with a specific antibody can be seen with the naked eye or quantified with an electronic plate reader. The reaction is less intense as the serum is diluted and the amount of antibody captured in the wells decreases. The titer is the highest dilution with definite color development. Many simple rapid diagnostic tests detect specific antigens in biological samples by using an enzyme immunoassay. The purpose of this animation is to explain how a prototype example of this assay works. The enzyme immunoassay can be done in a multi-well microtiter plate or on any other solid adherent surface. I will use the microtiter plate in this example, so let's take a closer look at one of the wells of this plate to see what happens during the performance of the assay. The plate is prepared to perform a particular assay by coating the wells with antibodies that bind to the antigen of interest. Then the wells are filled with the clinical sample, which could be a sample of serum, respiratory secretion, cerebrospinal fluid, urine, or some other body fluid. If the antigen is present in the sample, it will bind to the fixed antibodies. In this example, the green shape represents the antigen of interest, and the other shapes represent other molecules in the sample. However, note that only the specific, or green antigen, and none of the irrelevant molecules bind to the antibody-coated wells. This accounts for the specificity of the test. The wells are then washed out to remove any of the unattached molecules, leaving the antigen of interest stuck to the wells. Now, a second antibody, directed against another epitope on the target antigen, is added. These antibodies are conjugated covalently to an enzyme, indicated by the yellow circle at the Fc portion of the second antibody. They bind to the antigen that's fixed in the well, and this provides a second level of specificity for the assay. The wells are washed again to remove any unbound antibodies, and in the final step, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate and the captured enzyme generates visible color in the solution. At the macroscopic level, the development of color indicates those samples that have second antibody bound to the target antigen in the wells. Thus, the wells that change color are the ones that contain the antigen of interest. In other words, the positive tests.